All right. So last time we left off, we were talking about the lipid bilayers, and we had started talking about these lipid bilayers like so. You remember they have the two fatty acid chains with a glycerol and phosphate portion to the head right here, which looked like this. Okay, polar head right here, non-polar tails here, right? And the difference between the previous fatty acids and soaps was this had two tails, which gave it the bilayer with the nonpolar region here, and then the polar region facing out towards the water, right? Or in this case, it would be the interior contents of your cell and the exterior aqueous environment, what we tend to call like your blood and your intermembrane space. Okay? And that is you are giant bags of water. You know that, right? We all are. Okay. Okay, so what happens here is we're going to see that the lipid bilayer, that is your cell membrane, actually has a number of protein molecules. Some of these molecules can actually span the lipid bilayer, and these are called integral membrane proteins. They're going to be associated with one hydrophilic surface of your bilayer, and so that means they have to have. Excuse me. The other option is that they're attached to the surface of your lipid bilayer. So instead of running through your lipid bilayer, which is the integral, meaning through, they're going to be on the surface, meaning peripheral. So these are referred to as peripheral membrane proteins. So the proteins are going to be the membrane's functional components typically. They're going to allow things like selecting specific molecules to move through the membrane rather than them passively being moved. Okay. They can also take energy to move things through, so you can build up concentrations in your cells. Okay. They can do things like have little identifiers attached to them. They can go ahead and bind to a signaling molecule and then signal inside your cell to do something, like an insulin. Okay. So the exterior surface of your cell membrane is also going to contain your carbohydrates. These will act as your cell signals. That's that blood type, remember, where we had the carbohydrates that are embedded on the surface of your cell membranes. So the fluid mosaic model is going to have essentially floating pieces that float around within this fluid mosaic model, which is your lipid bilayer. Okay. Remember that your lipid bilayer continuously moves like a fluid. Right? It doesn't move super fast, but it still moves. So the easiest way to think about this, you can think about them like little icebergs that are floating around within the sea. That is your lipid bilayer, right? Now these icebergs can be mostly above water or they can be mostly below water, right? And that would be the cases that we see up here, where we have the integral, which would be mostly below water running through it, right? And then we have the above that's floating on the top, bound to the very top of it, which would be the peripheral. So remember that your membrane is going to be fluid-like. Your body's actually going to, not so carefully, but attempt to regulate the stiffness and fluid-like characters of your cell membrane. Okay? And it does that by adding things like cholesterol into your cell membranes and changing the lipid lengths and having cis and trans fats. Okay? So these would change the stiffness or fluidity of your lipid bilayer. So right here we see the phospholipid bilayer. Again, that's the hydrophilic on the outside. This would be your carbohydrate side chains bound to the top here. This would be an integral membrane protein. See how it goes through. Channel proteins are very, very common types of these. There are a few other ones that also are integral and transmit signals instead. They don't necessarily transmit molecules through. Okay. This right here is cholesterol. This is one of the ways you can regulate and change the stiffness of your membrane. Okay. If your membrane's too stiff, does anything know what would happen to it? Okay. Anybody ever used or seen different metals used in construction? Okay. So why don't they make irons harder than steel? So why don't we make buildings out of iron? Why don't we make like cars out of iron? Why don't we typically give you iron shears? Iron shears would actually cut better than steel ones. Or scissors. Why don't we do that? Because iron's very stiff and it's not very strong. Okay. And what that means is that iron, if you had like iron shears, they used to have a lot of problems where you'd actually, when they'd break, the chips would actually fly off and hit people. Okay. And so they actually break fairly commonly. You know how you take like a chip out of your scissors by accident because you cut something too hard? Iron does that a lot. And so like iron knives, nobody uses them because they break and chip all the time. Okay. That's what would happen if you had too stiff a membrane, right? When they're bouncing around in your blood system and getting whacked by things, they'd break. Now, if you have it too fluid, what happens? It just comes apart, right? And so you need to keep a fluidity and stiffness where it stays stiff enough to stay together well, and you sort of maximize your strength of your membrane, okay? Okay. This right here is your cell membrane, in case you forgot. This would be your cytoplasm within here, the cell membrane. And then you have right here the nucleus, which is another membrane in us, another lipid bilayer that then has the DNA within it, right? You also have some other structures, but I'll let biology take care of that one. Okay. Okay. Relating quite well to biology now, aren't we? All right. So, steroids and membranes. Steroids is the base cholesterol. And so, cholesterol is a steroid. Steroids are going to be lipids. They have a structure that will contain this four-membered fused ring, which is the steroid nucleus right here, which cholesterol is a derivation of. So all steroids have this nucleus right here. Okay. And notice it is a 6-member, 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 5-membered, right? Okay. All right. Enjoy the beauty of it. All the controls in your body right there, right? Okay. Now remember that when you look at these steroids, the different steroids are going to differ greatly from the structure of fatty acids and triglycerides. This looks nothing like a normal fatty acid, does it? It doesn't have a carboxylate head. It doesn't have the triglycerol part. It basically looks nothing like, right? Okay. And so it is actually derived from a fatty acid. Okay. But it's wrapped back around in on itself and bonded into this structure. Now, these are going to have a variety of molecular functions in your body. It will have things like regulating sexual development, which would be your testosterone and estrogen, emulsifying dietary fats, bile acids, and of course, we have things like signaling in your body to do various functions, okay? Which is actually the sexual development signals for testosterone and estrogens do that as well. So this right here is actually the cholesterol. See, the nucleus is right here. And then we have these groups, which are the difference from that. Okay. And so that's cholesterol. Note that it does have a polar end right here. And so cholesterol is going to situate itself into the phospholipid bilayer. And it has this nice flat structure here, right? Fairly flat structure. And it's pretty reinforced because it has two bonds along the structure. And so what you end up seeing is it actually acts as a sort of stiffener in it, okay? It acts as a little bridge that can connect multiple of the fatty acid chains. Right? So the OH group here is going to be what protrudes out into the aqueous environment. The rest of the molecule is going to nestle right here into that nonpolar layer, okay? Another language we can use here would be hydrophilic, right? And hydrophobic, okay? You knew that was going to come back to the lab information, didn't you? All right. So again, cholesterol is going to slip in between the saturated hydrocarbon tails, your phospholipids. It's going to change the fluidity. It'll actually, increase the fluidity because it will break up the nice overlayer. Okay, let me draw that out actually because I don't have a nice figure in here. Somebody stole. Okay, I'm going to draw it on the slide. Every time. Okay, so remember that a fatty acid chain looks like this. Okay, so normally the fatty acids that are saturated will overlap very nicely, right? Like kind of like a Pringles in a can, right? Okay. So this is actually going to look a bit like coming out towards you, away, towards you, away, and then this would come out towards you over here. So this is actually going to look a bit like this, but wide, right? And so it's like, has anybody gotten a Pringles can where they screwed up one of them? And it like curves into itself, and then it doesn't situate right, right? It's like a double wide instead of being the single, okay? It's going to end up like that. This is the double wide right here. And so it can increase your fluidity of your system. Now, if it interacts with the unsaturated tails, it's actually going to increase the rigidity of your system. So, and that's because the unsaturated tails have that little kink on them. Here. Like that. Okay? And so this will actually bind better to the unsaturated tails, whereas the saturated ones, it binds worse to. Okay? And so depending on what you have the most prevalence in that area, it will actually change your fluidity, increasing fluidity with saturated hydrocarbons, breaking them up, decreasing fluidity or increasing rigidity if you have the unsaturated ones. Is that making some sense? Okay. So, all right, so this right here is your study guide again, provided to you so you can print them off if you ever want to and go over the information. Anybody have any questions? Chapter 7. Okay. We wrapped back around to biology stuff, didn't we? Good thing.